Hello, my name is Idalis Boyd, and my partner's name is Alexis DePina from Biology 125, Section 4, Group 1. For our project aims, the cause of cancer is defects in the mismatched DNA system. However, the overall goal is to see how a mutation can affect a cell and the importance of a mismatch repair. So your question may be, how is this to happen? So we are to locate MSH2 gene and distinguish it from an empty vector type, mutant type, and wild type to be able to understand HNPCC in humans. Also, hereditary non-polyposis colon cancer. Colorectal cancer is also known as colon cancer. It starts as a polyp, a growth that starts in the inner lining of the colon or rectum and grows towards the center. There are two types, FAP and HNPCC, which we are going to dive into more in detail. Hereditary non-polyposis colon cancer results from genetic alterations of the DNA mismatch repair genes, most frequently in MLH1, MSH2, and MSH6. We will use the budding yeast because it can test for MSH2 function. The experimental design is the goal is to identify which mutation can affect the repair function of MSH2. So for the materials and methods, we began with 3 E. coli strands and isolate the plasmid using ZP Plasmid mini prep. We also distinguish an empty vector from PMSH2 by digesting 2 enzymes, APA1 and XBA1. We also ran the agarose gel to determine which plasmid DNA contains the MSH2 to insert. PCR, also known as polymerase chain reaction, was used to amplify and copy DNA sequences. We also diluted each plasmid to 5 nanograms per microliters using nuclease-free water, and also to determine the forward and reverse primers. We also digest the 3 PCR products using a restriction enzyme that can distinguish the mutant MSH2 from the wild type MSH2. We will also continue to further this experiment by doing yeast transformation, which will be used to identify the function of the MSH2 protein containing the H669Y, which was our mutation. We will also be using the FOA assay, which will be used to measure mutation rate in 3AGY75 yeast strands. This is a picture of the mini prep yield of DNA data, and the results shows the collection that we got, which correlates to the isolating plasmid DNA. Also, here you will see our agarose gel. You will see that this will be the 1 kb ladder, where this one will be the uncut, cut, uncut, cut, uncut. Wait, 1 kb ladder, uncut, cut, uncut, cut, uncut. So a huge error in our experiment was that we did not see any DNA that should have been in our cut samples, all 3 of them. So also we use 3 systems to help us identify a mutant. APE was used to select, find, and align our mutant wild type sequences so we could locate the mutation. Clustal was used to compare both sequences, which were translated to protein sequences. Clustal allowed us to locate the mutation in the protein sequence form of both the wild type and mutant. And the neb cutter was used to distinguish the enzyme that cuts the mutant and not the wild type. So here you will see the wild type PMSH2 in the neb cutter system. In the second one, you will see the mutant H669Y. The BMR1 is the enzyme that seems to cut only the mutant and not the wild type.